I'm Professor Sims, and in this video I will discuss the nervous system and nervous tissue. This is the last in a series of 10 lessons held as part of my Anatomy and Physiology 1 course. If you are a student currently enrolled in this course, please consult the syllabus and course Moodle site for assignments, quizzes, due dates, and other course information. In this unit, we will learn about the major divisions and functions of the nervous system, gray matter and white matter, the parts of the multipolar neuron in order of signal flow, the types of glial cells, their function, their association with the central nervous system or peripheral nervous system, resting potential, acting potentials, and graded potentials. And finally, we will learn about the major types of neurotransmitters. Anatomically, the nervous system is divided into the central nervous system, also known as CNS, and which includes the brain and spinal cord, and the peripheral nervous system, also known as the PNS, which comprises all of the cranial and spinal nerves that link the CNS to the rest of the body. The CNS serves as the control center, processing sensory information, coordinating motor response, and governing mental activities such as reasoning, memory, and emotion. The PNS is functionally divided based on function, right? The sensory, also known as afferent division, which transmits information from sensory receptors to the CNS, and the motor, also known as efferent division, which carries signals from the CNS to the effectors like muscles and glands. Sensory input originates in somatic receptors located in the skin, muscles, and joints, as well as in the visceral receptors within internal organs. The motor division includes both the somatic nervous system, also known as SNS, and the autonomic nervous system, also known as ANS. The SNS controls voluntary movements of skeletal muscles, while the ANS regulates involuntary activities in cardiac and smooth muscles and glands. Although somatic and autonomic pathways have distinct functions and targets, they often overlap, which is to say that a single stimulus can simultaneously trigger a conscious somatic response and an involuntary autonomic adjustment. The three major divisions of the ANS include sympathetic, parasympathetic, and the ENS. The sympathetic division is responsible for initiating the body's fight or flight response, increasing heart rate and respiratory rate while suppressing non-essential functions like digestion. In contrast, the parasympathetic division supports the rest and digest state by slowing the heart rate and stimulating digestive processes. The third division, the enteric nervous system, functions independently to control the gastrointestinal tract, regulating the activity of smooth muscle and glands involved in digestion. Although the ENS is atomically part of the PNS, it works closely with the autonomic system. Functionally, the nervous system performs three primary tasks. First, sensation involves detecting changes in the internal or external environment through specialized receptors and transmitting this information to the CNS. Second, integration occurs within the CNS where the sensory information is processed and interpreted in order to determine an appropriate response. Third, the system initiates a response by activating muscles or glands, thereby helping to maintain homeostasis. Let's watch a short video to help summarize what we just discussed and to help us to transition into some more complex topics about the nervous system.
You can divide the nervous system into two very general regions: the central nervous system, CNS, which consists of the brain and spinal cord, and peripheral nervous system, PNS, which consists of all other components of the nervous system, such as nerves throughout the body. The PNS can provide sensory information for the CNS. While the CNS can process that information and act as a command center, the CNS can execute motor responses or regulate body mechanisms. So we said the CNS consisted of the spinal cord and brain. Let's talk a little bit about the amazing human brain, although realize we're being very general here, as we're going to divide it into three general regions: the hindbrain, midbrain, and forebrain. Let's look at the hindbrain first. It includes the medulla, pons, and cerebellum. The medulla has many regulation functions, such as the regulation of breathing, blood pressure, and heart rate. The pons is involved with some of these functions as well, and also coordinating signals with this area to the rest of the brain. And the cerebellum? Balance and movement coordination are some functions of the cerebellum. The midbrain, deep in the brain, this area is involved in alertness and the sleep-wake cycle, motor activity, and more. If you've heard the term brainstem, this includes some of the structures we just mentioned, the medulla, pons, and midbrain specifically. Finally, the forebrain. Most notably, this includes the cerebrum, which itself is divided into two hemispheres, right and left. So many functions are done by our amazing cerebrum depending on specific location, whether it's our speech, our thinking and reasoning, our sensing, our emotions. Check out the further reading to explore this. The forebrain also technically includes some structures in it, like the thalamus, which is involved with sensory and motor information, and hypothalamus, which, if you remember from our endocrine system video, has major control of the endocrine system. There are a lot of myths about the brain. One quick myth I heard all the time as a kid that I have to put to rest. It's the myth that humans only use 10% of their brain. It's not correct. We have a great reading suggestion on that, as well as some others that circulate around. Now, that was all part of the central nervous system. What about the peripheral nervous system? Function-wise, we can further divide the PNS based on what it does. The somatic nervous system, SNS, and autonomic nervous system, ANS. The SNS is involved with motor functions of skeletal muscle. This will include voluntary actions under conscious control, but also somatic reflexes and involve skeletal muscle. The ANS is all about what's going on in the internal environment in regard to gastrointestinal or excretory or endocrine or smooth or cardiac muscle, and it also includes autonomic reflexes. And the ANS itself can be further divided. I know, there's a lot of dividing, but stay with me. The ANS can be divided into the sympathetic and parasympathetic systems. The sympathetic system, the shorter word of the two, helps you remember it's part of the quick fight or flight response. I know the whole running from a bear is a very popular example. For me, it'd be more relevant if I was face-to-face with my personal nemesis, the copy machine, which I may or may not have had some very bad experiences with before, and the bell that just rang so you know you have 60 seconds to get your copies, but it's making crazy machine noises and giving you vague warnings. This could also activate your fight or flight response, a response that can cause your heart to race and breathing rate to increase, and some things to not be as active, like the digestive system, because if you're desperately trying to run from a bear or take on the copy machine, you don't really need to be digesting food at that moment. Right. The parasympathetic system, longer word, this is often called rest and digest. Heart rate will decrease, digestion will occur, again, rest and digest. Many times these two systems can therefore have opposite effects on the same organ. So let's talk about two major types of cells in the nervous system that makes up nervous tissue. That means these are cells that you'll find in the central nervous system and the peripheral nervous system. Most of the time, neurons are what come to mind. There are different types of neurons, but to focus on general neuron structure, you have the cell body. Nucleus and most organelles are here. There are dendrites. Generally, these branch structures are where signals are received. And you have the axon. I like to think away axon because axons are the fiber where normally a signal will be carried away to some other cell. The junction area where the neuron will be communicating with another cell is called a synapse. And the other major cell type? Glial cells, or you can call them glia. When I was a student and read that they were supporting cells, I don't think the word supporting emphasized to me at the time how essential they really are. Structurally, there was a lot of emphasis on how they actually help the neurons connect in place. The word glia comes from a Greek word that means glue, but glia have huge roles, and they're so much more than that. Some glial cells keep a balance of certain chemicals in the space between cells, essential for signaling, and maintain the blood-brain barrier, which keeps a lot of substances in the body from getting into the nervous system. Some glial cells make myelin, which goes around the axons of neurons as something called a myelin sheath. It insulates the axon and the transferring of the signal. Some glial cells produce cerebrospinal fluid, which is protective to the brain and essential for homeostasis, as well as many other critical functions. Some glial cells have important immune function in the nervous system. These are all just a few examples.
Neurons are the functional units that receive, process, and transmit electrical signals. Gray matter contains neuron cell bodies and unmyelated fibers. It's involved in information processing and integration. White matter is made up of axons that transmit signals over long distances and oligodendrocytes that make up the myelin sheath. Both gray and white matter include a number of glial cells, which are often referred to as supporting cells of the CNS. The term glia comes from a Greek word meaning glue, referring to their structural role in holding neurons in place. Some glial cells help regulate the chemical environment between neurons and perform other functions that we will discuss shortly. The brain has three major regions: the hindbrain, midbrain, and forebrain. Hindbrain includes the medulla, pons, and cerebellum. The medulla regulates vital functions such as breathing, heart rate, and blood pressure. The pons supports similar functions and helps relay information between the brainstem and other regions of the brain. The cerebellum plays a role in maintaining balance and coordinating movement. The midbrain located deep within the brain, is also involved in regulating alertness, motor activity, sleep-awake cycle, and together the medulla, pons, and midbrain form the brain stem, which is responsible for basic life-sustaining functions. The forebrain, again, includes the cerebrum, which is divided into right and left hemisphere and is responsible for higher-order functions such as thinking, reasoning, speech, emotion, sensory perception. It includes the thalamus, which is the relay center for the sensory and motor signals, and the hypothalamus, which is involved in autonomic regulation and control of the endocrine system. Neurons are the excitable, long-lived, and typically non-dividing cells responsible for transmitting electrical signals throughout the body. They are among the largest and most specialized cells in the human body. Although there are different types of neurons, their general structure includes three main parts: the cell body, or soma, which contains the nucleus, rough ER, also known as nysal bodies, and most of the cell's organelles, serving as the neuron's metabolic center. Dendrites are branched extensions which receive incoming signals and conduct them towards the soma. And then the axon begins at the axon hillock, or the trigger zone, and it's a long fiber that generates and transmits action potentials away from the cell body. Axons terminate in the telodendria and axon terminals, which contain synaptic vesicles filled with neurotransmitters. The point where a neuron communicates with another cell, whether another neuron, muscle fiber, or gland, is called a synapse, and the gap between two communicating cells or neurons is called the synaptic cleft. A presynaptic neuron is the nerve cell that sends a signal across a synapse, and a postsynaptic neuron is the cell that receives and responds to that signal. Many axons are wrapped in a myelin sheath, which increases the speed of signal transmission. In the peripheral nervous system, Schwann cells form the myelin sheath, while in the central nervous system, oligodendrocytes form the myelin sheath. Myelinated fibers conduct signals rapidly and make up the white matter, whereas unmyelinated fibers conduct more slowly and form the gray matter of the brain. Nodes of Renvier are the gaps between segments of the myelin sheath, and these help propagate the action potential along the axon. Loss or damage of myelin, known as demyelination, is associated with disorders such as multiple sclerosis.
Multiple sclerosis is a demyelinating disease of the central nervous system, which includes the brain and the spinal cord. Myelin is the protective sheath that surrounds the axon of neurons, allowing them to quickly send electrical impulses. This myelin is produced by oligodendrocytes, which are a group of cells that support neurons. In multiple sclerosis, demyelination happens when the immune system inappropriately attacks and destroys the myelin, which makes communications between neurons break down, ultimately leading to all sorts of sensory, motor, and cognitive problems. Now, the brain, including the neurons in the brain, is protected from things in the blood by the blood-brain barrier, which only lets certain molecules and cells through from the blood. For immune cells like T and B cells, that means they have to have the right ligand or surface molecule to get through the blood-brain barrier. This is kind of like having a VIP pass to get into an exclusive club. Once a T cell makes its way in, it can get activated by something it encounters. In the case of multiple sclerosis, it's activated by myelin. Once the T cell gets activated, it changes the blood-brain barrier cells to express more receptors, and this allows immune cells to more easily bind and get in. It's kind of like bribing the bouncer to let in a lot of people. Now, multiple sclerosis is a type 4 hypersensitivity reaction, or a cell-mediated hypersensitivity. And this means that those myelin-specific T cells release cytokines like interleukin-1, interleukin-6, tumor necrosis factor alpha, and interferon gamma, which together dilate the blood vessels, which allows more immune cells to get in, as well as directly cause damage to the oligodendrocytes. These cytokines also attract B cells and macrophages as part of the inflammatory reaction. Those B cells start to make antibodies that mark the myelin sheath proteins, and then the macrophages use those antibody markers to engulf and destroy the oligodendrocytes. Without oligodendrocytes, there's no myelin to cover the neurons, and this leaves behind areas of scar tissue, also called plaques or sclera. In multiple sclerosis, these immune attacks typically happen in bouts. In other words, an autoimmune attack on the oligodendrocytes might happen, and then regulatory T cells will come in to inhibit or calm down the other immune cells, leading to a reduction in the inflammation. Early on in multiple sclerosis, the oligodendrocytes will heal and extend out new myelin to cover the neurons, which is a process called remyelination. Unfortunately, though, over time as the oligodendrocytes die off, the remyelination stops, and the damage becomes irreversible with the loss of axons.
Neurons can also be classified by structure, and axonic neurons have indistinguishable axons and dendrites and are found mainly in the brain and sensory organs, though their function remains unclear. Bipolar neurons have one dendrite and one axon. They are rare and they're found in the retina and nasal mucosa. Unipolar neurons have a single fused axon and dendrite, and they're common among sensory neurons in the peripheral nervous system. Multipolar neurons with one axon and multiple dendrites are the most common type of neuron, and they are included in all motor neurons. Specialized neuron types are also present in the nervous system. Pyramidal cells are multipolar neurons found in the cerebral cortex. They're shaped like pyramids, and they're involved in voluntary motor control. Purkinje cells are located in the cerebellum, and they have highly branched dendritic trees, and they coordinate motor movement. Olfactory neurons are responsible for detecting smells, and as you can see, these are very different in structure than the other neurons that we've seen. Glial cells greatly outnumber neurons, with an estimated 700 to 900 glia per neuron. Their type and function differs between the CNS and PNS, as we've seen with the other structures of the nervous system. In the CNS, astrocytes are star-shaped cells that help regulate the extracellular environment, recycle neurotransmitters, form scar tissue after injury, and maintain blood-brain barrier. Microglia act as immune cells by engulfing pathogens and cellular debris. Oligodendrocytes produce myelin sheaths around multiple axons. Ependymal cells, which line the ventricles of the brain and central canal of the spinal cord, produce and circulate cerebrospinal fluid using their cilia. In the PNS, Schwann cells myelinate individual axons. The outermost layer of the Schwann cell is called neurolema, and the spaces between adjacent Schwann cells are the nodes of Ranvier, which we've seen in the diagram of the myelin sheath. Satellite cells, which surround neuron cell bodies in the ganglia, regulate the chemical environment in a manner similar to the astrocytes, though they do not contribute to a blood-brain barrier. Figures 12.11 and 12.12 show the different types of glial cells found in the central nervous system and the peripheral nervous system that we just discussed. Sensory receptors detect stimuli and convert them into electrical signals, which travel along different sensory neurons, typically unipolar or bipolar, to the central nervous system. Interneurons, which are usually multipolar or axonic, process and interpret the incoming information, allowing for integration, decision-making, and the formulation of appropriate response. Once a response is determined, motor neurons, which are generally multipolar, transmit signals from the CNS to effectors, such as muscles or glands, resulting in actions like muscle contraction or gland secretion that enable the body to respond effectively to the stimulus. Sensory input begins when a sensory receptor, such as the thermoreceptor in the skin, detects a stimulus like a change in water temperature. This detection activates the sensory ending, generating an electrical signal that travels along the sensory neuron's axon towards the spinal cord. At the spinal cord, the signal is transmitted across a synapse by converting the electrical signal into a chemical signal, which then triggers a new electrical impulse in the next neuron. This signal travels along the sensory pathway to the brain, passing through the thalamus and ultimately reaching the cerebral cortex, where the temperature is consciously perceived. After integrating the sensory information with other neural inputs, the brain initiates a motor response by sending a command back to the spinal cord. The motor pathway involves two neurons: the upper motor neuron, which originates in the cerebral cortex and synapses in the spinal cord, and then the lower motor neuron, which extends its axon from the spinal cord to a skeletal muscle. At the neuromuscular junction, the signal causes the muscle to contract, completing the body's response to the original stimulus. Nerve cells, or neurons, maintain a resting membrane potential, which refers to the difference in electrical charge across the cell membrane between the inside and outside of a cell. In a typical diagram like the one shown here, the extracellular fluid is shown above the cytoplasm, and it is separated by the plasma membrane, which consists of a phospholipid bilayer. To measure the electrical potential difference across the membrane, a voltmeter is used. One electrode is placed inside inside the neuron and another outside the neuron, and the voltmeter typically reads around negative 70 millivolts, indicating that the interior of the neuron is more negatively charged than the exterior. This negative resting potential is mainly maintained by the sodium-potassium pump. This is a membrane protein that actively transports 3 sodium ions out of the cell and 2 potassium ions into the cell. Since more positive charges are pumped out than brought in, the inside of the neuron remains negatively charged. At rest, there is a high concentration of sodium outside the cell and a low concentration inside, while potassium is more concentrated inside the cell than outside of the cell. Additionally, negatively charged proteins, which cannot cross the membrane, are abundant inside the neuron and further contribute to the internal negative charge. There are several types of ion channels in the membrane, each serving a specific function. Passive ion channels or leakage channels are always open and allow certain ions to move across the membrane. These channels may selectively restrict ion flow based on size exclusion or electrochemical charge. For instance, a channel may attract oppositely charged ions, or it might be too narrow for larger ions to pass through. Gated ion channels open only in response to specific stimuli. For example, ligand-gated channels open when a signaling molecule, such as a neurotransmitter, binds to them. These channels are found on dendrites and the soma, and they play a key role in generating graded potentials. Voltage-gated channels respond to changes in membrane potential and are important for initiating and propagating action potentials. They are located along the axon. Action potentials are electrical signals generated by ion movement across the neuron membranes. An action potential operates under 2 important principles. In myelinated neurons, the action potential jumps between the nodes of Ranvier, the gaps in the myelin sheath, in a rapid process called saltatory conduction, and this greatly increases the speed of transmission. The action potential follows the all or none principle. Once the membrane reaches threshold, the signal fires completely, much like flipping a switch. If the threshold is not reached, no action potential occurs. As we've discussed, the neuron maintains a polarized membrane when at rest. This is the resting membrane potential. When a neurotransmitter binds to the ligand-gated sodium channels, sodium enters the neuron, causing depolarization, a shift in membrane charge toward 0. This creates a graded potential that spreads across the dendrites and soma. If the depolarization reaches the threshold of negative 55 millivolts at the axon hillock, it triggers an action potential that travels down the axon to the synaptic terminals. After the action potential peaks, sodium channels close and potassium channels open, allowing potassium to exit the cell. This outflow of positive ions repolarizes the membrane, restoring a negative internal charge. The membrane may temporarily become more negative than its resting state, resulting in hyperpolarization, which is usually around negative 90 millivolts. And during this refractory period, the neuron cannot generate another action potential until the sodium-potassium pump restores the original ionic balance. The sodium-potassium ion pump actively transports 3 sodium ions out of the cell and 2 potassium ions into the cell, and this helps to reestablish the original ion concentration and return the membrane potential to approximately negative 70 millivolts. Here's a clip that helps tie together the concepts of the ion-gated channels, action potentials, polarization, depolarization, repolarization, and hyperpolarization.
Neurons are the cells that make up our nervous system, and they're made up of 3 main parts. The dendrite, which are little branches off the neuron that receive signals from other neurons, the soma, or cell body, which has all the neuron's main organelles like the nucleus, and the axon, which is intermittently wrapped in fatty myelin. Those dendrites receive signals from other neurons via neurotransmitters, which, when they bind to receptors on the dendrite, act as a chemical signal. That binding opens ion channels that allow charged ions to flow in and out of the cell, converting the chemical signal into an electrical signal. Since a single neuron can have a ton of dendrites receiving input, if the combined effect of multiple dendrites changes the overall charge of the cell enough, then it triggers an action potential, which is an electrical signal that races down the axon up to 100 meters per second, triggering the release of neurotransmitter on the other end, and further relaying the signal. So neurons use neurotransmitters as a signal to communicate with each other, but they use action potentials to propagate that signal within the cell. Some of these neurons can be very long, especially ones that go from the spinal cord to the toes. So the movement of this electrical signal within the cell is super important. But why does a cell have an electrical charge in the first place? Well, it's based on different concentrations of ions on the inside versus the outside of the cell. Generally speaking, there are more Na+ or sodium ions, Cl- or chloride ions, and Ca2+ or calcium ions on the outside of the cell, and more K+ or potassium ion, and A-, which we just use for negatively charged anions, on the inside of the cell. Overall, the distribution of these ions gives the cell a net negative charge of close to negative 65 millivolts relative to the outside environment. And this is the neuron's resting membrane potential. When a neurotransmitter binds to a receptor on the dendrite, a ligand-gated ion channel opens up to allow certain ions to flow in, depending on the channel. Ligand-gated literally means that the gate responds to a ligand, which in this case is a neurotransmitter. So let's take the example of a ligand-gated sodium ion channel, which when it opens lets sodium flow into the cell. The extra positive charge that flows in makes the cell less negative, since remember it's usually negative 65 millivolts, and therefore less polar, so that's why gaining positive charge is called depolarization. Neurotransmitters typically open various ligand-gated ion channels all at once, so ions like sodium and calcium might flow in, while other ions like potassium might flow out, which would actually mean some positive charge leaves the cell. In the end though, when it's all added up, if there's a net influx of positive charge, then it's called an excitatory postsynaptic potential, or EPSP. In contrast, the opening of only ligand-gated chloride ion channels would cause a net influx of negative charge, creating an inhibitory postsynaptic potential, or IPSP, making the cell potential more negative, or repolarizing it. Now, a single EPSP, or IPSP, causes only a small change on the resting membrane potential. But if there are enough EPSPs across multiple sites on the dendrites, then collectively they can push the membrane potential to a specific threshold value, typically around negative 55 millivolts, although this can vary depending on the tissue. When it hits this threshold value, it triggers the opening of voltage-gated sodium channels at the start of the axon, which is called the axon hillock. These voltage-gated channels open in response to a change in voltage, and when these open, sodium rushes into the cell. The influx of sodium ions and the resulting change in membrane potential causes nearby voltage-gated sodium channels to open as well, setting off this chain reaction that continues down the entire length of the axon, which ends up being our action potential. And when this happens, we say that the neuron has fired. Once a lot of sodium has rushed across the neuronal membrane, the cell actually becomes positively charged relative to the external environment, up to about positive 40 millivolts. This depolarization process ends when the sodium channel stops allowing sodium to flow into the cells, which is a process called inactivation. But this inactivated state's different from when the channel's closed, or open for that matter, which are the two states that most other channels have. The voltage-gated sodium channel, though, is unique in that it has what's known as the inactivation gate, which blocks sodium influx shortly after depolarization, and stays in this state until the cell repolarizes and the channel enters the closed state again, and the inactivation gate stops blocking influx. Even though that inactivation gate's not blocking it anymore, the channel's still closed, so no sodium enters the cell in this state. The middle open state's therefore the only state where sodium gets let into the cell through the channel, and this is a very short window of time. Now, in addition to these sodium voltage-gated channels, we've also got potassium voltage-gated channels, which are slow to respond and don't open until the sodium channels have already opened and become inactivated. The result of this is that after that initial sodium rush into the cell, potassium flows out of the cell down its own electrochemical gradient, removing some positive charge and blunting the effect of the sodium depolarization. These potassium channels don't have a separate inactivation gate, and therefore they stay open for slightly longer, which means that there's a period of time when there's net movement of positive ions out of the cell, which causes the membrane potential to become more negative, or repolarize. During this repolarization phase, the cell also relies on the sodium-potassium pump, which is an active transporter that moves 3 sodium ions out of the cell and 2 potassium ions into the cell. It's during this repolarization phase that the cell's in its absolute refractory period, since the sodium channels are inactivated and won't respond to any amount of stimuli. This absolute refractory period keeps the action potentials from happening too close together in time, and also keeps the action potential moving in one direction. The combined efforts of this pump and the extended opening of the potassium channels result in a small period of overcorrection, where the neuron becomes hyperpolarized relative to the resting potential. During this time, the sodium channels go back to their initial closed state, and for a short period, the potassium channels stay open. At this point, we're in the relative refractory period, since the sodium channels are closed, but they can be activated. Although because the potassium channels are still open and we're in a hyperpolarized state, it takes a stronger stimulus to do so. Finally, as the potassium channels close, the neuron returns to its resting membrane potential.
Graded potentials occur in the dendrites of the neurons and may be either depolarizing or hyperpolarizing. These local changes in membrane potential vary in strength and can summate to influence whether a neuron reaches the threshold needed to initiate an action potential. Summation can occur in two ways: spatial summation, where multiple presynaptic neurons contribute to the stimulus, and temporal summation, where a single presynaptic neuron fires repeatedly over time. Graded potentials arise from different sources. A generator potential occurs when a sensory stimulus directly stimulates the dendrites of a unipolar sensory neuron, such as those in the skin, and can lead directly to an action potential in the axon. A receptor potential occurs in specialized sensory cells, such as those involved in taste, vision, or hearing, and typically results in the release of neurotransmitters onto adjacent neurons. When graded potentials are generated at synapses, they're referred to as postsynaptic potentials, PSPs. Excitatory postsynaptic potentials, or excitatory PSPs, depolarize the postsynaptic membrane, increasing the likelihood of an action potential, while inhibitory PSPs, or IPSPs, hyperpolarize the membrane, making an action potential less likely. Neurons communicate at synapses, which can be either electrical or chemical. Electrical synapses are formed by gap junctions, and they allow ions to pass directly between cells, enabling rapid signal transmission. While found in both the central and peripheral nervous systems, electrical synapses are relatively rare. Chemical synapses are far more common, and they involve the release of neurotransmitters from the presynaptic neuron into the synaptic cleft. These chemical messengers then bind to receptors on the postsynaptical neuron, initiating a cellular response. The presynaptic neuron sends the signal while the postsynaptic neuron receives it. The effect of a neurotransmitter depends not on the molecule itself, but on the type of receptor that it binds to. There are two major types of receptors that determine the postsynaptic response. Ionotropic receptors, such as nicotinic acetylcholine or glycine receptors, are ligand-gated ion channels that produce immediate short-term responses by allowing ions to flow directly into the cell. In contrast, metabotropic receptors activate intracellular signaling pathways through G proteins and second messengers, producing slower but longer-lasting effects. Neurotransmitters are chemically diverse and fall into several key classes. The cholinergic system uses acetylcholine, which functions as at the neuromuscular junctions and in certain areas of the nervous system. Amino acid neurotransmitters include glutamate, which is excitatory, as well as glycine and GABA, which are inhibitory. Biogenic amines, such as dopamine, serotonin, norepinephrine, and epinephrine, are derived from modified amino acids. Neuropeptides, such as vasoactive intestinal peptide and gastric inhibitory peptide, are composed of amino acid chains and modulate neuron activity over longer periods. Please watch this fun animation that gives just a few examples of neurotransmitters that work in the brain.
Our brain consists of billions of neurons, specialized cells which communicate with each other. Neurotransmitters are small molecules that transmit signals from one neuron to another across the synapse. In this way, they control all our feelings and emotions. There are different neurotransmitters, each having different tasks. This, for example, is GABA. By binding to his receptor, he creates the feeling of calmness and helps us to relax. This is melatonin, a lazy guy, always sleeping on his synapse. He's very active every night, makes us feel tired, and helps us to sleep. But now's not the right time to be tired. Wow! Now it's time for oxytocin. Oh, what's going on? It's adrenaline, a very stressed and frightened molecule, even afraid of jumping out of a synapse. Come on, Adrenaline, relax. It's not a scary situation. Maybe our friend GABA can help you calm down a little bit. Thank you, GABA. Now it's time to enjoy love. And time for dopamine to spread happiness all over the brain.
This concludes the material for Lesson 10 and for this course in general. As always, thank you for watching. Don't forget to do the reading, check the description for more videos related to these topics, and leave your questions for me in the comments below.